Everybody to Wednesday Night Lab. I'm Tom Zinnan. I work at the UW-Madison Biotechnology Center. I also work for the Division of Extension. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night by Zoom, 50 times a year. Tonight it's my pleasure to introduce to you Allison Bender and Caryn Wadler. They both work at the Wisconsin Energy Institute and the Great Lakes Bioenergy Research Center. They're going to be talking with us about an update on biofuels and other stuff that they can synthesize in the processes of making biofuels beyond biofuels. So this is going to be good stuff. I think Allison is going to go first. So I'm going to start with Caryn. Caryn, where were you born?
I'm from the Chicago suburbs.
Where'd you go to high school?
I went to Libertyville High School.
Oh, and then where'd you go for your undergrad?
Champaign-Urbana, still
What'd
Illinois.
you study?
I studied microbiology and biology.
Very good.
And literature.
What kind of literature?
Just English literature.
Good. And then where'd you go for your master's or PhD?
I did a master's also in Champaign-Urbana, and then I came here to UW-Madison for my PhD.
And that was in microbiology?
Yes, I was in Garrett Suin's lab in microbiology.
Very nice. And now you're a postdoc with?
With Michael Thomas.
Very good. Thank you. And then Allison.
Hi.
Where were you born?
I was born right in Madison, Wisconsin.
And where'd you grow up?
In Milton, Wisconsin.
Was that
about an hour south.
Yeah. And is that where you went to high school?
Yep, Milton High School.
And where'd you go for your undergrad?
I went to St. Olaf College in Northfield, Minnesota.
And then what'd you do?
And then I completed an AmeriCorps year working as a naturalist in a state park in southeastern Minnesota. And then I got hired after that year here at UW-Madison. So just entering my third full year of my job here at Madison.
Excellent. Well, it's a pleasure to have both of you. And Allison, if you'd like to start, I want to welcome both of you to Wednesday Night at the Lab, and Allison Bender is going to lead off for us.
Yeah, thank you so much for having us. I'm excited to be with you all virtually today and to share this talk with Caryn. I am the outreach coordinator at the Wisconsin Energy Institute and the Great Lakes Bioenergy Research Center. So what I'm going to do for you guys today is give a really high-level overview of what it is we do at the Research Center and then pass it over to Caryn so she can dive into some of her science for you all. So the Great Lakes Bioenergy Research Center is a large Department of Energy funded grant with over 400 scientists and staff who are all working on different pieces of this puzzle to transform biomass, or in our case, plant matter, into fuels and products that would otherwise be made from petroleum or oil. We are administratively housed at the Wisconsin Energy Institute at UW-Madison's campus. You can see the Energy Institute on the map here, just about a block away from Camp Randall Stadium. The Energy Institute is the home for all sorts of energy-related research that's cross-disciplinary on campus, so everything from the biofuels to microgrids. And I also wanted to note that the Energy Institute, as well as all of UW-Madison, is located on traditional Ho-Chunk land. The Ho-Chunk people have been here since time immemorial and call this area J-Jope. And we want to recognize that their inherent sovereignty and vibrancy remains strong in Wisconsin, and along with the 11 other First Nations in Wisconsin. And it's also important for me to mention that tribal nations and indigenous peoples worldwide are doing some of the most important and leading work in transitioning us away from fossil fuels to clean energy systems. So back to Great Lakes Bioenergy. This is our mission statement here. We work to develop economically viable and environmentally sustainable biofuels and bioproducts made from dedicated energy crops on marginal lands. So I'm going to break that down in the next 15 or so minutes as we go through this slideshow together. We are not the only Department of Energy funded research center who are working on this puzzle. You can see 3 others on the slide here. We're all led by large universities or national laboratories. And so there's a lot of brilliant people who are working on this challenge of how do we convert plants into fuels and products. So you might think, and you're right, that there's a lot of motivation for doing this. I'm just going to list a couple examples here today. First, if we were to have a fully realized bioeconomy making biofuels and bioproducts, we would be supporting Wisconsin's rural economy, providing jobs in particular for farmers and for manufacturing. Secondly, you might be aware that Wisconsin doesn't have any fossil fuel reserves. We import all our coal, our oil, our natural gas. So if we were to use our plentiful pastures and forests and what we do have to grow and produce and manufacture our own products, we would be supporting our own energy independence. And then last but not least, there are a myriad of different environmental reasons why we would want to make the switch from petroleum-based products to bio-based products that would enhance our soil and water health, support biodiversity, and not least to mention, reduce carbon emissions and mitigate climate change. Because that last reason is so big, and because 2020 has been a year like none other for seeing some of the detrimental impacts of climate change, I wanted to pull us back just for a moment and talk about what is going on with global climate change. So you can see this graphic on the left here. We've got our Earth and our atmosphere made up of greenhouse gases like carbon dioxide and methane and water vapor. And we've got the solar radiation, that heat and light coming in from the sun, warming the Earth, and then some of that bounces back off the surface of the Earth. Some of that leaves through the atmosphere, and some is captured and re-emitted back down towards the atmosphere. Sorry, towards the Earth. And is this a problem? Absolutely not. This is what keeps our planet livable for everything on the planet. But what is the problem is when we humans have taken ancient sequestered carbon dioxide, so hundreds of millions of years ago, that plankton and that algae that lived in the ocean and then settled on the bottom of the ocean surface when it died over millions of years, was compressed into the coal, oil, gas, the fossil fuels that we use today. When we extract those and burn them, we're emitting what used to be sequestered ancient carbon, and we're emitting them into the atmosphere. So you can see it's sort of like throwing another heat trapping blanket. Sorry for the phone ringing in the background. I think my family will get that soon. Apologies for that. So it's like another heat trapping blanket captured and re-emitting heat. You can see this arrow here is a little bit larger, sending that captured heat back down onto the Earth. So where are these greenhouse gases coming from? This is a graph from the EPA, from greenhouse gas emissions in the US in 2018. And we can see that the largest section of that is transportation, 28%. So even as we are electrifying our cars and trucks, we still have jet planes and cargo ships that need a really high energy intensive fuel. And we need to figure out a way to decarbonize those transportation options. So I pose this question. What if we could make a fuel out of something that is removing carbon dioxide from the atmosphere? And you might be thinking the answer already. Plants obviously can do this. How do they do it? Photosynthesis. So throw back to your high school science class. You can see the equation for photosynthesis here. We've got carbon dioxide in the atmosphere being converted into the sugars, into the glucose that makes up the structures of the plant. So when we think about biofuels, we're thinking about the vision of having plants growing and taking in carbon dioxide from the atmosphere equal to the amount of carbon dioxide that, yes, indeed, is released when we burn those plants that have been converted into fuel. And it's not just those two pieces of the puzzle. You can see the cycle on this slide here, obviously very simplified, but from the harvesting to the transportation to the processing, all of those parts need to be equal. Any emissions that are coming from those processes need to be equal to what the plants take in when they're growing. An even loftier vision beyond carbon neutral is having a carbon negative situation, where we imagine the plants being perhaps amazing prairie plants with deep roots that are, as those plants grow and those root systems grow, that stay in the ground holding and sequestering that carbon, that perhaps is a greater amount of carbon than all the other processes to make and to use the fuel or the product. So that's sort of our vision as we think about biofuels. I also wanted to address this question before I got any further, because you might be thinking of ethanol. Ethanol is in nearly all of the gasoline that we use today, but I wanted to mention because our center actually currently doesn't work on corn or ethanol. There are a couple of reasons why, but one of those is what we call the food versus fuel problem. We don't want to take any land or resources that would be originally dedicated to providing food for people or animals and use that for bioenergy. So you might remember in our mission statement, we mentioned marginal lands. The land, the agricultural area that we use to study and to grow our crops would be land that is no longer suitable for growing food. Maybe it doesn't yield as well, it's depleted, maybe it's too hilly. For a variety of reasons, that's the land that we want to focus on, because perhaps that land could grow switchgrass or mixed prairie, and that could be useful for bioenergy. Secondly, we are moving away from annual cropping systems towards perennial systems that don't need to be replanted year after year. By their very nature, many of these perennial cropping systems are less energy and resource intensive. They're more resilient to drought, perhaps need less nutrient inputs. And so there's a variety of different reasons why we would focus on that as well. And then lastly, as a government-funded research center, our job is to really push the understanding. So we're not working on corn ethanol because it's already scaled and it's already kind of figured out. We're working on new questions and new fuels and new products that all the questions aren't figured out yet, and there's still a lot of challenge in making it a reality. So you can see here some of our research plots. These are at Kellogg Biological Research Station in Michigan. Some of the crops that we do study here, we've got mixed prairie, we've got switchgrass, another native perennial prairie grass, and poplar trees. So ideally something that's native and perennial and low input are characteristics that we're studying. And researchers are working on many different pieces of how do we grow these crops sustainably without adding nutrients or water, and how do the microbes on the leaves and the roots interact. So there's a bunch of different research teams who are all studying things with crops in the field. This graphic is showing our pipeline at the research center. And after all I spoke about, we're still only at the very start of it. So I'm going to go through the rest in a really high level way, just give you a taste of some of the questions our researchers are asking with this work. So once we've got the plants, the next step is to break down the plant into the part that we're really looking for to make biofuels and bioproducts out of, and that's the cellulose. So you can see this graphic of the plant cell wall. We've got the cellulose in yellow. And the challenge here is that it's all tangled up and tied up in other parts of the cell wall, the lignin and the hemicellulose. So the very first step is to break down that cell wall with the goal of gaining access to those sugars and to that cellulose. This pre-treatment process can look like a lot of different things. It could be heat or acid or pressure, adding ammonia, or adding salts, what we call ionic liquids. After we've broken down the plant cell wall, then we use enzymes to sort of cut that cellulose up, that chain of glucose molecules, into the digestible pieces that we would then pass on to the fermentation. So that's kind of the middle step, taking the plant matter, breaking it down into the usable sections, and then on to fermentation. So this over here is a picture of what we call hydrolysate. That's the broken down plant matter that we would essentially feed to a microbe, either bacteria or a yeast, to do the work of fermenting that into the product that we're looking for. Down here on the bottom is a picture of Saccharomyces cerevisiae. That's a yeast that you might have some familiarity with. Humans have had a relationship with this yeast for thousands of years, using it to rise bread and to make beer and wine, and it's actually one of our most common lab microbes. So one of the cool research projects that's been ongoing, this is Trey Sato, one of our scientists. He and a team of researchers have been scanning through different yeasts to figure out which yeasts are more tolerant of some of those chemicals from the last step. So you remember salts and acids and heats, all those things that we use to break down the plant can be toxic to the yeast that does the fermenting. So they've been looking at different strains of yeast and kind of identifying what genes make a yeast tolerant to that sort of situation so it can ferment most efficiently. Another kind of cool project is working with a different strain of wild yeast that produce naturally this red pigment you can kind of see here in the center. And that red pigment is created in the same biochemical pathway as isobutanol is. And isobutanol is another fuel that is of great interest to our research center because it is more energy intensive than ethanol, it's less corrosive than ethanol, and can be blended into gasoline at any percentage. So it's a really desirable product. And we're studying sort of what this yeast already does, what it's good at doing naturally, and how can we use that part of its DNA to produce the products that we want it to. Okay, so there we are at the biofuels and bioproducts. And I'm really glad that Caryn is here because this graphic doesn't even show the true end of our system. So after the fermentation, there's gunk, conversion residue left over, and Caryn's portion of the talk is going to give you a little bit more of a taste of what happens after this fermentation. But one of the beautiful things of the center is that we've got all these different experts, from agronomists in the field to people who study enzymes to engineers to people who do the techno-economic analysis to make sure it all makes sense. We've got all these different pieces of the puzzle working together to make a system that potentially could be scaled and could be viable in the real world, in the real marketplace. And one story that I like to think about that kind of shows how crucial having all these different parts of the system talking together is a couple of years ago, our researchers who were doing the fermentation were working on a hydrolysate that had been passed to them from other researchers downstream, and it didn't produce the results that they were expecting. And they were able to go back and look at the experiments that had been done and the data that had been collected about that particular hydrolysate, and we're able to identify that that switchgrass was grown in 2012 in Michigan, where there was a large drought. And because of that, the switchgrass grew differently, and the yeast weren't able to ferment the hydrolysate in the way that they had typically been able to in a non-drought year. And so because of the communication and the collaboration amongst different disciplines and different research projects that we have at our center, our researchers were able to figure that out and are now expecting a drought into how can we deal with things like that will come up when you're growing plants in the real world, and how can all these different pieces work together. So I'm going to leave you with this graphic of sustainability as 3 different factors: environmental, economic, and social. And just a couple pictures, recognizing there are a lot of different ways that we work towards sustainability. And I am really glad to be in community with you all as we each kind of take our own path towards that greater goal. So thank you so much for your attention. Apologies for the interruption with the phone there, and I will pass it over to Caryn, and I'll be open to questions after she's done with her part of the presentation.
Thanks, Allison. I think that was really great. Has it switched over?
Looks good.
Okay. So my talk is going to be, like Allison said, about what happens kind of at the end of this pipeline. So just to bring that back up, the way that the center works so amazingly together is that even though there's many different groups of researchers, we're all kind of working on getting things from this starting point where there's plant biomass to biofuel, and we're working on making it most efficient because we're doing it all in collaboration. So the people who are developing the marginal lands are telling the people who are engineering the plants what the plants need to be able to grow on, and the people who are looking at how the plants are broken down are looking at what the microbes need to eat, and so they can say, make sure when you're doing this, and so they can kind of do everything together to make sure that it's optimized for the next step, and that when they're designing it with all of those different pieces in mind. And so what we wanted to do was kind of add on to this biofuel story. And there's a whole bunch of stuff, carbon still left over after you make the biofuel. And like Allison said, we call that conversion residue. And so our thought was, could we get more out of this plant material by taking this conversion residue after you remove the biofuel and subjecting it to another round of microbial fermentation? So having bacteria try to take that leftover stuff and turn it into something else that we could use. And this idea kind of came from the petroleum industry, because one of the reasons that petroleum is so cost effective as a fuel is because not just because it all makes fuel. Like you can see from this graph, about half of the barrel of crude oil gets turned into gasoline, but a whole bunch of other stuff gets turned into products like plastics and other things that we use. So we're like, okay, maybe we can use more of the plant material, just like the way the petroleum industry uses more of their barrel that they get of crude oil. So some of the challenges that we started thinking about when we were doing this were, we're working at the end of the pipeline. That means that the material that we get isn't always going to be the same. And as they keep optimizing steps earlier in the process, what we get and what we're working with might be different. So we have to think about getting what we get, and that we're going to have to modify how we use it depending on what we get. So we need to think of a system that's flexible enough to work with whatever material might be left over after all of the biofuel is removed. There's also shifting economics and markets. When we think about what bioproducts we're going to make from this conversion residue, we have to keep in mind that what's really valuable today might not be what's really valuable to make tomorrow. And there could always be a shift in the market where all of a sudden, oh my gosh, the lemon crop failed. We need a lot of a product like limonene to make our pine sol things smell lemony fresh. And how can we address these kind of minute changes that might come up at a moment's notice? Like, how are we able to make bioproducts that are valuable now and will be valuable years from now? And then thirdly, we're working with microbes. So whenever you engineer microbes, you can either take kind of one of two approaches. One is you take a microbe that's really easy to work with in the lab and try to get it to do that thing that you want it to do, whether or not it normally can. And then kind of the second strategy is taking a microbe that already does what you want it to do and try to get it to do that better. And there are pros and cons to both of these different strategies. So when we were taking into account all of these different challenges, that we need a microbe that's going to be flexible enough that we can engineer it to do what we want and make the different products and eat a bunch of different substrates, we kind of came across this organism called Streptomyces that we decided to start working with for our research. So Streptomyces is a soil microbe, and it undergoes this unique lifestyle where it starts off from a spore, and then that spore germinates and grows out from where it starts using these mycelium and hyphae. And then once it runs out of resources, it can go back into the spore, and then the cycle can go again as it keeps using more different resources. And so why did we want to work with Streptomyces? Well, because Streptomyces grows in the soil, it encounters a whole bunch of different material, such as the sorts of materials you might expect when you break down plants. And so it can grow on a bunch of different carbon sources, so many different compounds which we would expect to be in this conversion residue. And I'll talk more about that later. It has a well-developed genetic system that you can use between different strains and species. So that goes along with that engineering, that there's a bunch of things that we can do with it because there's already systems in place to get it to do the sorts of things we might want it to do if it can't already. And so just what I mean by that is the Streptomyces genome is a little bit unusual compared to other microbes. So this here is a graphical representation of a Streptomyces chromosome. And most of the time when you see a bacterial chromosome, it's a complete circle. But you can see the Streptomyces has these two little blue dots on top. So it actually has a linear chromosome. And what that means is that the chromosome is kind of divided into two parts. And so if you can see, there's kind of this dark blue center. I don't know whether mouse is working. Is this? Yes, there we go. Has this dark blue area kind of at the center of this linear chromosome. And that's called the core. And it has central metabolism and these things called primary metabolites. And then it's got these arms, which are kind of at the edge of the chromosome. And so it produces these things called secondary metabolites. And Streptomyces has been used industrially to make these secondary metabolites. And so when we do engineering with Streptomyces, we can engineer things on the edges of this chromosome here which means it'll be less likely to affect central metabolism and primary metabolites and all the things that the cell needs to grow, which is convenience in terms of engineering for it. So my talk is going to focus on two different things: how we looked at Streptomyces' ability to use and grow this conversion residue, and then how it makes bioproducts. So first, I want to focus on looking at Streptomyces' ability to grow and utilize this conversion residue. So one of the advantages of working with Streptomyces is there's a whole bunch of different Streptomyces out there. And this here is a phylogenetic tree for Streptomyces. And the blue lines are members of the tree that are called type strains. And so those are ones that are well known. And this phylogenetic tree works a lot like a family tree for bacteria. So just like on a family tree, the closer the branches are, the more related they are. And then the farther apart they are, the less closely related they are. So you can see we have this wide range of Streptomyces, and they are very different. So our hope is we have a whole bunch of these different strains to use. Some of them are going to be able to grow on this conversion residue. And the black lines represent this collection of Streptomyces that we got from another lab called the Curry lab. And they were able to isolate these from nature. So these are natural isolates. So I just took that tree and kind of flattened it out to make it a little easier to see. And those are some of the different strains that we're working with. Each of those lines represents just a different strain of Streptomyces. And the first thing we did was put these strains into some conversion residue and said, can they grow? Will they use this? And the answer was yes, some of them will. And so I've highlighted with black boxes all of the ones that were able to grow on conversion residue. And it wound up being actually about half of the collection. I only have a subset that I'm showing here on this tree. And so my question was, well, this is really interesting. We have a bunch of Streptomyces that can grow and use conversion residue, but we also have ones that don't. And why might that be? So we had to do a little analysis on what exactly is in conversion residue. We've talked about how it's this stuff left over after you make biofuels, but what does that mean? So here is a bottle of conversion residue. Allison was totally right. It's this sludgy black tar stuff that is left over after you remove biofuels. And this is just a bottle that I got from the Biotech Center. And we did a whole bunch of analysis on it. And some of the things that we identified, I say we, it was the Biotech Center entirely, were xylose, carbohydrates, acetamide, which is left over from the breakdown of plant material processing that they do, glycerol, acetic acid, which is a waste byproduct from the zymomonas that initially turned this into biofuel, proteins, which are again from the zymomonas that died making our biofuel, ethanol, another byproduct, toxic aromatics, those are mostly from the initial breakdown of the plant material. They're lignin-derived compounds usually, and so they're from that stuff that was kind of between the cellulose that was in that picture earlier. And it's actually fairly acidic at a pH of 5.8. Normal average would be about 7 for bacterial growth. Now, the interesting part is, even though we know all those things that are in there, there's still 70% of that stuff that we don't exactly know what it is. We know some of it is cellulose, but cellulose actually can be a whole bunch of different lengths. So we don't know how big the cellulose is or how small, and that's just a lot of stuff that we don't know what it is. So to kind of give you an idea of what that looks like visually, these are the known components. There's glucose, xylose, pyruvate. These are just the ones that we're able to identify with HPLC analysis. And you can see that there's a good range of byproducts left over from the zymomonas, and even things like glucose, cell bios, xylose that were products from the plant material that the zymomonas wasn't able to utilize. So it wasn't able to eat all of it. There's still some left over for our microbes to work with. But in terms of the entire percentage of the conversion residue, there's still that big green part, which is all that cellulose and hemicellulose and unknown components that we have to work with. So one of the things we wanted to do is try to understand how our Streptomyces were growing on this conversion residue. So we took the conversion residue, we grew our Streptomyces, and then we did the same analysis that we used here to determine what was in the conversion residue to determine what components our Streptomyces were eating. And when we did that, for each of our different strains, we got a whole lot of data, which is really great when you're a scientist because yay, information, but doesn't always mean that there's answers. So in this graph here, you can see this green component is all of that unknown again. And if you look far on the very right, this is just conversion residue. So this is the starting material. And you can see here, like some of these strains, we're able to eat about a third of it. And that's some of our best eaters. They're able to eat a bunch of this unknown, but they were also able to eat a bunch of the known components. Like this one was able to utilize a lot of the xylitol. And some of the other strains were able to utilize, say, a lot of the xylose. So what we learned here is that not all of these Streptomyces strains are going to use the conversion residue in the same way. And that's really interesting and useful because it means in the future, if we get different conversion residues, we can say, oh, that conversion residue has a lot of xylose. This strain here is really good at utilizing xylose. Let's use that strain to use that conversion residue for different byproducts. Alternately, we can do engineering and try to make a master strain that can utilize all of the different things as best as they can. So this gives us a really great springboard in terms of engineering future strains using components from these strains, or just using these strains and optimizing it for whatever we get, which is really great considering one of those challenges is we don't know exactly what the conversion residue of the future will be, since we were talking about sci-fi earlier, and who knows what the future may bring. This also kind of showed us some patterns. Like there's definitely some strains that seem to prioritize utilizing this unknown component of the conversion residue over the known component. And so that's something that I find really interesting and kind of want to study those. Why do those strains go after this unknown, which is cellulose or hemicellulose, which you might traditionally think of as being harder to access instead of, say, glucose, which you could just import and put directly through your central metabolism. So it starts getting at kind of these basic science questions as well as these questions for engineering things later. So the second part of my talk here, I want to talk about how we get Streptomyces to make terpenes. So earlier we had talked about getting Streptomyces to make bioproducts, and the bioproducts that we kind of hit upon in terms of the economic analysis were these things called terpenes. And this is a secondary metabolite. And interestingly, Streptomyces actually already makes a variety of terpenoids. So from an engineering perspective, this is really great. One of the problems that was run into earlier with biofuels when they were trying to make bioethanol was they were using E. coli to make it, and E. coli didn't normally make bioethanol. And so as E. coli made more and more ethanol, the cells would die because it wasn't resistant to it. And you kind of ran into this issue that the more of the product that was made, the less the cell could handle it. Since Streptomyces already makes these products, that means hopefully we wouldn't run into these issues of, oh no, I made too much, and then the cell can't make any more because it has hurt itself. Now, some of these compounds you guys may already be familiar with. Some of these are used industrially as antibiotics or antifungals. One of the ones that a lot of people know about and are very familiar with is geosmin, which is that aroma that you get right after it rains, that kind of wet earth smell. And so also, if anyone does any source water contamination issues, methyl isoborneol is one of those compounds that is a source water contaminant. And so that's one of the things that they look for there. So all of these terpenoids look very different, but they're actually made via the same basic pathway. And so how that pathway works is it comes directly off of central metabolism through this thing called the MEP pathway. And you get these two products, DMAP and IPP. And so DMAP kind of acts as your anchor, and then you add a molecule of IPP on kind of in this stepwise way. So you go from this 5-carbon compound to this 10-carbon compound this 15 carbon compound to this 20 carbon compound. So each time you add another 5 carbons, making a bigger and bigger chain, which you can then modify to make all of those different compounds that you were seeing before. And you can do that either by directly modifying it into one of the compounds with a synthase. So if this 5 carbon compound can become isoprene, which then could become isobutanol if you were making fuels or something. You can get all of these different 10 carbon compounds, pinene and limonene, which are things that are used in scents and different fragrances. You can get bisabolene, which is another fuel, farnesol, I think is another fuel, and these other 15 carbon compounds. Or you can then start combining these 10, 15, and 20 carbon molecules to make even bigger isoprenoids. So you can take 2 15 carbons, put them together, and you can get the 30 carbon squalene, which is used in makeups as a moisturizer oil. Or you can take 2 of these 20 carbon compounds, put them together, and get carotenoids such as beta carotene, which is in your carrots, and lycopene, which is in your tomatoes. So we talked about all these compounds, and we know that Streptomyces can make them. So what engineering needs to be done? Put these guys in a thing of conversion residue and watch the dollars roll in, right? Well, that'd be fabulous. But Streptomyces doesn't always make all of these compounds all the time. And so you can see here you have this Streptomyces avermidilus and this Streptomyces lividans, and they're growing on this plate, and they're growing in white light. And they found out recently, I think it was just a couple of years ago, that in order to get these guys to make lycopene, which was that compound down at the end, that 40 carbon terpenoid, you actually have to expose them to blue light. And so if you put blue light on this plate, all of a sudden this avermidilus is making lycopene. It's turning red, just like you would expect a tomato would be, like that color red. But how are you going to be exposing these guys to blue light in a lab setting in a giant fermenter vat? Like, that's just not very feasible. So we wanted to go about coming up with a way to engineer our strains to make these terpenoids all the time so that we could have them make what we want as much as we want. So the first of these many terpenes that we decided to engineer is actually lycopene. As Allison was mentioning earlier, it turns your cells this bright red color, so it's very easy to see if your engineering efforts have worked or not. If it's red, it worked. If it's not red, it didn't work. And it's also at the very, very end of this pathway. So kind of logically, if it can make lycopene, it can make every single one of these other compounds on the way to lycopene. So that way we'll know that if it can do this, we can engineer it to make these other ones as well. So we actually came up with this way of engineering the plasmids using a system called Golden Gate cloning. So Golden Gate cloning is very useful when you want to make a bunch of different things. And so we were trying to come up with a strategy that we could use because we knew that we're not just going to be making lycopene. We're going to want to make other things eventually. And depending on how the economics and the markets and everything change, we're going to need to be able to rapidly change what these bugs are producing. So the way Golden Gate cloning works is that there are these enzymes that cut DNA, and they recognize a certain sequence of DNA, and then they cut. Now, most restriction enzymes, they recognize the DNA and they cut right where they recognize. But these enzymes are special in that they recognize the sequence here, but then they cut whatever is 4 base pairs down from it, no matter what it is. So that means that we don't have to worry about the way that the site cuts affecting the gene we want to put in there, because we can have this sequence and then we can put in any gene we want here. Or then we could use the sequence and put in a second gene or a third gene, because some of these products need multiple genes to make them. So for instance, we started with this plasmid here, where we have the genes necessary to make lycopene, these CRT EIB. And this is a modified promoter region. So this is going to make sure that these genes are always on. They're not going to need blue light or any other weird way to make them. And then we have this other plasmid, which has these special enzymes to put these genes on the chromosome of the Streptomyces we want, and an antibiotic resistance so that we can check to make sure that we have the plasmid that we need. And it has this gene here that will turn cells blue. So if we haven't moved these genes over, the cells will be blue. And if we have moved these genes over like we want, the cells will be red. And this is a very convenient way to just look at cells and know whether or not you have what you want, instead of having to go through the effort of checking your DNA and doing a whole bunch of analysis. So made this plasmid, put them in the Streptomyces, and lo and behold, we were able to turn them red. Now, just like we saw before when we looked at different Streptomyces and saw that there were different ways that they utilized the conversion residue, there's also different ways that these strains make lycopene, different amounts that they can make. So on all of these plates, I have the initial strain that I started with, and then the plasmid to make lycopene. And you can see that some of them were able to make a lot of lycopene, turn the cells really red, but some of them were only able to make a very little bit of lycopene and turn them kind of this light pink. So this was all very exciting. I was able to engineer cells, get them to produce lycopene, but what are they going to do when I introduce them to conversion residue? When you add conversion residue to cells, they're going to utilize them differently. Does that also mean that they're going to make different amounts of lycopene like they do on this media, or is something going to happen? Are we even going to see lycopene at all? So I took all of those different strains that you were seeing earlier, grew them in conversion residue, and then spun them down, and good news, I was able to see red. So these strains are able to grow on conversion residue and produce lycopene, which is what we were hoping for. So you can see this guy here on the right is some extract from one of the lycopene producing strains, and it's red. And this is an extract from a strain, the same strain that did not produce lycopene, and it is this yellow-brown color that is normal color for it. So I was able to show that the Streptomyces were able to ferment conversion residue through this pathway to make lycopene. And so now I'm going to be moving on and doing different engineering things to make different of these terpenoids based on an economic analysis that we got previously, and see how I can engineer them and what different sorts of things we can make, which is very exciting. And so that brings us kind of back here, back to our pathway, and we're able to now add on this area of taking conversion residue and use Streptomyces to turn it into terpenes. And it's a really nice way to kind of get more out of this pathway. So I just want to take a few minutes and acknowledge all the many, many people in the GLBRC that make this research possible. I work for Michael Thomas in the Thomas Lab with a postdoc, Kurt, who has been doing all of this stuff with me. And Curry Lab was the one that gave us all of the strains that we were using for the engineering and the original collection that we're working with. And then the Hittinger, Noguera, and Amador Nuguez lab also work in the same group as me, engineering microbes to make different bioproducts. They do not work with Streptomyces, but we work hand in hand, kind of doing things to look at how our organisms utilize conversion residue and then what the best products are for our organisms to make. So we've been doing a lot of protocols with them and trying to see if there are ways we can work together to use more of these stuff. Core facilities make all that conversion residue for us and do that HPLC analysis with the metabolites like I was showing earlier. And then we were able to do more of the conversion residue analysis over using these facilities at MSU. I've talked to Sean Mansfield at University of British Columbia about other conversion residue components we can add. Christos does our economic analysis, and then Trey, we heard about earlier, he does a lot of the work in the center as well. So with that, if anyone has any questions, I know Allison and I are ready for them.
Great, thank you very much. Kendall, do you have any in the chat, or do you want me to read the questions in the chat?
There was some in the chat, but I think Allison responded to them.
Okay.
So
Does anybody have any questions they'd like to address to either
Yes.
Allison or to Caryn?
You're starting out using type culture microbes, bacteria, Streptomyces in your case, and you know the things that wind up being type culture organisms are abnormal in the natural sense, in that most of bacteria seem to be living in association with other bacteria and or fungi. And the question is this the most efficient way of trying to make use of this byproduct? In other words, might it make more sense to go out to nature and try and find naturally occurring associations of microorganisms which could make better use of the residue that you have? I know it's a more complex thing to do, and it's not as, you don't have the unique genetic engineering quite as readily available for that, but it might wind up being a way of finding something that could work more effectively and perhaps ultimately be more tunable to a range of products.
So that is exactly what the Noguera lab is doing. One of those other labs that I was showing in the acknowledgement, they have wastewater communities that they've isolated, and they're using those to grow on the conversion residue as well. And so our hope is that when they have the communities growing, they'll be able to identify genetic elements that help them utilize different things. And then we can use that when we're engineering Streptomyces to try to, so that way we can kind of combine our efforts. They can find communities that will give us more material to work with in terms of things that might be helpful for utilizing, but then we can engineer things that will produce exactly what we want. And so that's exactly what we're doing.
Thank you.
Other questions? So I'd be interested, it's we're celebrating the 40th anniversary of the Bayh-Dole Act, which is about patents. Caryn, can you tell me what the status is of any of your research as far as patenting goes, and how you go about, how does the GLBRC and the Wisconsin Energy Institute go about patenting? And then do you do any of the licensing, or is there another licensing agent that GLBRC works with?
That might be an Allison question. I have never done any patenting or licensing. I had to sign an NDA to use some of the materials that we get. But
What's an NDA?
A non-disclosure agreement.
Oh, so if you get stuff from somebody else, you agree?
There's a proprietary enzyme cocktail that's used to break down the initial plant material for the first use it. So since the conversion residue might still have some of those enzymes in it, in order to work with the conversion residue, I had to sign an agreement that I would not let anyone else ever touch the conversion residue or do analysis to figure out what enzymes might still be in there.
Very good. Allison, do you have anything for us on patents and such?
Yeah, I just pulled up a graphic on my screen. I think this is not perhaps the most recent graph because it cuts off at 2019 there, but you can see some of the patent applications and licenses that have gone through since GLBRC has been funded. We've been a center since 2008, and we do use WRF to work through for our patents. And I didn't mention this, but GLBRC has scientists at multiple universities across the United States and Canada. And so I think that our partner universities, primarily Michigan State, we use the patent office that's associated with their university as well.
Yeah, that's a lot of patents and licensing there, this triple figures of licensing, and it's quite a portfolio. Other questions? Mark Schultz. Hi, Mark.
Yeah, I'm wondering, you know, this was a wonderful, fascinating scientific exercise, looking at all these biochemical pathways. But in order to make this of economic significance, you have to purify the components that have some value, and then hopefully select the ones with the best market. At your organization, has there been any economic study of the various outputs from this type of process?
Caryn, do you want to tell about how that one example of flooding the market?
Yeah, so Christos does the economic analysis that I mentioned on the acknowledgement slide. I can bring Christos Marvelius. I can bring him back up if you need his name. But he has published a couple different analyses, and I have worked with members of his lab who have come to me and asked about which methods we're using exactly. Like they want to know exactly how much we adjust the pH, how many mils of HCl we're going to be using per liter of compound, how much time it takes us to grow them, how fast we spin the shakers, because that's electricity that they're going to need. There's a very in-depth analysis at every step, like how do you purify it and what are you going to do? We make lycopene because it was an easy molecule to test, but like Allison said, when we told them we were making lycopene, they said, okay, well, you can't actually make a giant fermenter of lycopene because you will flood the market. That's just not a thing. Do not do that. Please do not do that. And so that's become code in our lab. When we've had a bad day, it's like, hey, you guys just want to make a giant reactor of lycopene? Let's just flood the market. I'm done with this research today. It's been so rough, so rough. But they have identified a bunch of targets that, like isoprene, they have told us that the market can bear as much isoprene as you can make. Just make it, yes, it'll be hard to purify, but go for that. And a couple of other ones that we're targeting as well. So the ones that we chose kind of for our second generation of production that I'm working on now are the ones that are supposed to be the most economically valuable.
There's a couple of questions in the chat. One's from Michelle Goulet and others from Dareth and Todd. Michelle asked, what's the potential market for terpenes? Have you costed out the production of terpenes using this technology versus current industrial processes?
So since I haven't put them in a giant reactor yet to see how much we can get, we don't know exactly how much we're going to have, but where's that question again? I'll bring it back up. The potential market is pretty vast. For some of the aromatics, like I was saying, like pinene and limonene, those can be used as food additives, as scent additives. Squalene can be used in makeup. Some of the, like bisabolene, I think is a jet fuel or can be turned into jet fuel. So the markets are anywhere from food industry, makeup fuels, and I think some of them can even be turned into plastics. There are a bunch of other terpenes that I didn't list, like we saw on the slide where I had the ones with like geosmin. They can be antifungals, antibiotics. There are some anti-cancer drugs in there. The center is focused on energy and not biomedical, so that's not something that we go for specifically, but if it's valuable enough, then it's a reasonable thing to make.
Great. And Dareth, did that cover your question, Dareth and Todd? Good. Other questions? Another question on GLBRC. How far are you into your most current contractor grant from the Department of Energy? Have you got 2 years, 5 years?
We are on year 3, I believe, of a 5-year cycle.
Okay. I bet those are intriguing documents to put together.
There's a lot of work that goes into putting together the annual documents and the quarterly reports, and every January we have a yearly review, and that's a big process. So that's coming up. People are starting to prepare.
And how is a complicated place, organization like GLBRC that has 2 lead universities, Michigan State and UW-Madison, several 5, 10 other places. How are you managing all this now that you can't meet together? What are your major strategies for coordinating all these things?
I think, Caryn, you can add after, but I feel like GLBRC was in a place to transition pretty well to this virtual world because we are spread across multiple universities. Our scientists were used to meeting across teams using virtual platforms, and so some of those systems were already in place. Since March, there's been a few, there was a sustainability meeting and other times where our scientists have gathered for online conferences and sharing presentations, and I think that it hasn't been that much of a challenge because people are used to having to have set processes for communicating when our labs are in different spaces, not just different buildings on campus, but different states and different universities. I don't know, Caryn, if you want to add to that.
I really don't have to. I think that it's gone really well. I've been really impressed with the conferences, actually. A lot of scientific conferences, there's walking around and looking at posters and talking with people, and a lot of the technology that they've implemented has been really amazing. I felt I got more out of the last virtual poster session that I went to than the physical one. I definitely had comfier shoes on.
All right. Other questions? All right. I see the director of the GLBRC, Tim Donahue, is here. I didn't know if Tim wanted to chime in on that last question or not. But thanks for coming in and listening, Tim. I appreciate it. Good. That's about everything for me. I'm going to turn the recording off, and then if we want to do any of the linger at the lectern where you can chat one-on-one with Caryn or with Allison, you can. I just have to find this.